And let me begin by thanking the organizers for the wonderful invitation to explore this new seminar format. Today, I would like to talk about B virus, which of course is a zoonotic virus that invades the central nervous system. But I would like to begin by telling you a little bit about Elizabeth Griffin, Beth Griffin. I never met, I had the fortune to meet Beth in life, but I did meet her in death. And at the time she had was a student at Agnes Scott College, which is an all-women's college here in Atlanta, an outstanding college for liberal arts and science majors. And Beth had, I believe, graduated and was trying to decide between medical school and graduate school. And she decided to take a position at Yerkes Primate Center to better understand research and see what a life in research was. And she had given her parents a tour the week before and showed them the colonies that belong to Yerkes and the Centers for Disease Control, and explained to them all about this endemic virus that the animals carried, and told them great stories about it and how dangerous it was. So it was somewhat amazing that within a week she had sustained an ice flash. And this happened in 1997, and she reported that she was informed by the supervisor that people are splashed all the time and not to worry about it. So this commenced a long journey that you know how it ended. Beth was a dancer. She was a free spirit. She was a scholar. She was intensely interested in medicine, biomedical research, and curing diseases. And she had a great deal of enthusiasm and love for her life and life in general. And so it was under these extraordinary circumstances that I met her, and I would like to dedicate this talk to her, as well as to the others who have lost their lives during my professional career: Steve Woodson, Larry Smith, Tom McGeorge, Dr. Bill Pollock, Elizabeth, of course, and 32 other known survivors that we follow in the laboratory. And so it's with looking towards these individuals as our teachers that I will present the data I have today to share with you what our current understanding of B virus infections are. And I'd like to remind everybody that although a very dramatic infection, it's a very low frequency infection with very high consequences. So fortunately, it does not happen often, but it happens, and it happens with some periodicity, and you'll see that as we move through the slides. The natural host of this infection, of course, are macaques. And why do we study this particular virus? Macaques have many viruses. There are still 15 to 20,000 macaques imported into the U.S. yearly. And I'll show you a little bit in the next slide of what the impact of that this past year has been. It's the most widely used non-human primate for research. It is also the endemic host of B virus. Rarely do B virus infected macaques have any overt signs of illness, but they do shed virus periodically, and perhaps we can come back to this point because there is some new knowledge that virus shedding, though not to external sites, may occur more frequently than we previously thought. It is the only known simplex virus in the herpes family that is zoonotic, and its transmission to humans frequently results in the death of humans that are not treated in a timely fashion. So I decided that I needed to know current stats and discovered to my surprise that importation of macaques has been on the uprise since about 2012. We're up 20% this year. We used to import around 30 to 40,000 macaques a year, but we're to 20,000, 19,000 in 2013, 17,000 in 2012, and as you can see, up to 23,000 monkeys and apes. And what kinds do we import? Well, we basically import a lot of fascicularis, far less macaca mulatta, and some of the other subspecies as well. And just for a brief review, I want to remind you that there are 3 kinds of herpes viruses: alpha, beta, and gamma. Alpha, the simplex viruses fall into the alpha category. And I bring this up because I frequently get the question, can I sample blood to discover whether an animal is infected? Can you tell me if there's virus in blood? Because B virus is an alpha herpes virus, it travels by route of the peripheral nerves. You won't find this virus in blood, unlike beta and gamma herpes viruses, which travel by the hematological route. So this is an important thing to remember when pursuing discovery of whether someone is infected or not. So as I mentioned, B virus is an alpha herpes virus of the simplex kind. You all are very familiar with alpha herpes viruses, whether you know it or not. Herpes simplex is, of course, the main representative of the simplex viruses, and varicella is the main representative of the varicella viruses. So there are simplex viruses and varicella viruses in humans. B virus is definitely a simplex virus. It's very closely related to HSV1 and HSV2. It has a very large genome, and that's important for us because that tells us that the virus has lots of genes for lots of different occasions. The primary infection at the point of inoculation of a person or an animal is at the epithelial fibroblast layers in the skin. So in the dermal epidermal region, we have cells that are very supportive of B virus replication. And these cells enable the virus load to grow in a rather short period of time so that the virus can then enter the peripheral nerves. So the virus travels via the peripheral nervous system to the dorsal root ganglia, and it goes anywhere from 16 to 30 centimeters a day, depending on how efficient transport is. While it's traveling in this course, acyclovir is not going to really do anything, nor is ganciclovir, because this virus now is not replicating en route to where it is going to set up housekeeping. It establishes a latent infection in the dorsal root ganglia, unless it goes beyond the dorsal root ganglia, and we'll talk about that in a minute. Reactivation means that the virus can come from the DRG back down the peripheral nerves that it originally traveled through, and begin replication again in the dermal epidermal layers of the skin. So that gives you a little bit of the complex pathways that the virus can take. We all know that macaques have few to no symptoms, but what we don't get to see very often, fortunately, is that humans suffer a mortality of up to 80%, and that can occur anywhere from 3 weeks post-infection to decades later in the absence of any previous symptoms. And what you can see at this point is very difficult, but I'm pointing my cursor at this place that seems to be a beige remnant of where the spinal cord used to be, where the brainstem enters the cranium. This is all that's left in acute cases of B virus infection. The upper spinal cord and brainstem simply liquefy. And part of that is due to virus replication, and part of it is due to the damage that the virus creates with respect to the kind of supportive care that patients need. B virus is a CDC risk group 4 agent. That means there is no cure, and there is a high mortality, high morbidity. Antivirals are only effective early in infection and preventing reactivation. And the virus can reactivate in surviving humans at any point in time. And because of that, risk group 4 agents the recommendation nationally in the U.S. as well as in other countries is that B virus be handled in a BSL-4 laboratory for the safety of those that are working with the virus. This is one of the cases from 1987. Most cases, the death is caused by respiratory paralysis and withdrawal of life support, or from infections that are acquired during this state. The CAT scans are usually performed early on and show a great deal of herniation. And interestingly enough, one of the signs of an infection is frequently at a very advanced state, the development of sinusitis, where virus causes immense draining of the sinus cavity, as well as other problems like diabetes insipidus. So I want to briefly take you through, we're not going to linger on this, but the first case was described in the early 1930s. William Brebner was a young physician who died as a result of working with pathologic tissues from a macaque. The next case wasn't noticed until 1949, and that was from rhesus saliva putatively to a patient's wound. Next case, 56. Then a series of cases between 57, 58, and those largely corresponded to the development of the polio vaccine and the massive importation of macaques into this country. So we saw periodic cases through the decade from 60s to 70s. Again, another case seemed to be isolated in 81. Then we had 4 cases in 1987, 3 cases in 1989, a series of cases through the 90s, and our most recent 2 cases occurred in 2015. Now, of the ones that perished as a result, I showed you those who had 5 fatalities since 1987. And that's a very large number for an occupational hazard. And because of that, it's imperative that we figure out ways to prevent future infections. One of the reasons why infections go untreated for an extended period of time is because physicians are often reluctant to consider B virus as one of the differentials of the diagnostic routine. It is the zebra in the room. It's not the usual thing to put on your differential diagnostic list. Symptoms that present are common to many different types of infections and are often flu-like. And there's rarely a time when there's not some type of flu going on. Also, although this is diminishing, there is an unawareness of B virus. It's frequently confused with hepatitis B, and so it becomes imperative that you go out and educate people that B virus is a separate virus and a very deadly one at that, and one that's of occupational concern. There is generally a lack of education, but as I mentioned, this is improving. So the result is that we have an unrecognized zoonosis until it's too late to do anything about it. When we do identify it, we rely on the detection of specific early and late antibodies. And the detection of virus or DNA is a rare event, but we can find virus under extraordinary circumstances, and certainly we can generally find the presence of virus in autopsy specimens and certainly in animal necropsies. Differentiation of early versus late antibodies by avidity assays also helps us tell approximately when a patient was infected. Is this an old infection? Is this a new infection? Not much more narrow than that can we isolate the time of infection. To give you an example, this particular individual that was followed at this point in time over 10 years was bitten in 1980. And over a period of time, you can see, this is months after initial diagnosis, you can see that there's waxing and waning antibodies. All the different color lines represent the assay that was performed. The navy blue represents the standard ELISA Western blot assay that is performed using virus. in trying to change that and make the assay more readily available for multiple labs, we designed and cloned and expressed different B-virus recombinant proteins, but you can see they're not quite as effective. The magenta is glycoprotein B, the yellow is glycoprotein D, and the green is glycoprotein G. We still use these as an additional test, but still the most sensitive test is working with B virus, either purified or in the infected cell lysates. Now, the other conundrum that we see is that people try to stratify the types of wounds that are most likely to have the possibility of virus contaminating them. But what we soon learned early on is that the wound, the severity of the wound has nothing to do with the ability of virus to get in. A mere scratch or a deep bite is equally effective at getting virus in. And the reason for that, as I best understand it, is to look at this cross-section of the skin. The virus enters usually epidermis or dermal layers, with bites definitely dermal layers. And remember, both of these cell types are very supportive for virus replication. The virus can expand itself, or it may be a very low load of virus, in which case it gets into the dermal epidermal layer and comes into contact with these nerve endings that then give it access to the axons that will then lead to the serving dorsal root ganglia. So let's look at that in a little more detail. Here we see a schematic, and if you'll follow with me, the virus enters, it can replicate in the dermal layers, and then at that point it can enter or access these sensory nerve endings, and at that point it can travel up the axon all the way to the cell body of the axon. So we call this retrograde, traveling away from the site into the cell body. And at that point, as I mentioned earlier, the virus can travel centimeters per day. And at this point in humans, most often the virus doesn't stop here. In macaques, it does stop here. And when it stops here, it can, of course, reactivate under stressful conditions and go back out to the skin source and replicate and freshen up the supply of virus that gets to the dorsal root ganglia. You see another schematic of this in the natural host from the top. The virus goes retrograde, from the entry site retrograde to the cell body, and it stops at the sensory dorsal root ganglia, which is still part of the peripheral nervous system, until it reactivates again and goes back out to these skin sites. And at that point, an animal can shed virus and inadvertently infect a human that's coming into contact with that animal. Now, I want to point out something that I just learned from some new data out of Larry Corey's lab, is that actually this process occurs much more frequently than we previously thought. But when it does, it can occur even weekly. Very little to no virus actually is shed or released. Instead, there's some very low-level dermal replication, and then the fresh virus goes back to the sensory ganglia and refreshes what population exists there. In humans, what we're most familiar with is that the virus bypasses this sensory ganglia and goes up into the spinal cord and lands in the brainstem, where it sets up housekeeping. So let's take a look at what that looks like. The virus, as it comes in, is going to enter the horn of the spinal cord. Let's look at this picture. It's a little bit more illustrative. The virus is coming in from the skin, ends up in the dorsal root ganglia, and then uses the sensory nerves to get into the horn of the spinal cord. You can see in a little more detail here that you have multiple nerves that can enter into the spinal cord And you can see a little more effectively in this slide that there are multiple layers of receptors that can become engaged by virus that's entering into this area. So we have visceral stimuli, we have nociceptor pathways and mechanoreceptors, temperature receptors. So we get a variety of reports from patients of the types of stimuli they feel once virus enters this area of the cord. From this point of entry, the virus can then travel upwards along these nerves that are going to ultimately culminate in the brainstem. And this schematic gives us a little bit of an overview if you're bitten on the hand, if you're bitten on the leg, if you're bitten on the torso or the face, which spinal ganglia are going to serve as the route of entry to the spinal cord. And again, you can see this from all these perspectives of the dermatomes where the virus is going to enter the spinal cord. And when it does enter the spinal cord, again, it's going to come in and react with all or one or two of these areas of the cord. And at this point, we can have one or two things happen. So if the virus comes into the cord at this point, it can travel straight up the cord. Oops, I'm going to go back to that one. Didn't mean to do that. It can travel straight up on the side of entry. But when it reaches this area called the dorsal column nuclei, it can then cross over and affect the brainstem on one side. And actually, we can even go up and find virus, not very much, but we can find virus in the cerebral cortex if the infection has been persistent for a long enough period of time. Or alternatively, the virus can cross over at the point of entry, in which case it still ends up on the same side, but what we see at postmortem is the other side of the cord is affected from the bite site or the point of entry. So this gives us a little bit of a picture of how the virus is trafficking from the leg, from the torso, from the hand, from the mouth or face, and then from the eyes. And so there are different sites of entry, but by and large, the virus is ending up in the same place in all cases. And again, this gives us a little bit better picture of the kinds of symptoms that we may see with different patients, whether it's numbness, tingling, hiccups, nausea, vomiting, etc. So this gives me a perspective of the kinds of pathways that the virus takes, and it gives us potential targets to study to see if there are any ways that we can develop to stop the course of the virus as it traverses to the brainstem. So the brainstem serves as the interface with the brain and the spinal cord, of course. It really controls respiration, and we see cessation of respiration. We see cardiovascular issues of low blood pressure. The sleep and wakefulness cycle is not dramatically affected, but gait control most certainly is, as well as sensory modulation. So let's take a look at what that's going to look like in the brainstem. We have basically three functions of the brainstem. And from our cranial nerves, for example, if the virus was transmitted by an eye splash, the cranial nerves are also going to feed into the brainstem. So it's a consistent target. What we see in postmortem tissues is that the pons, the medulla as well, the pyramidal tracts, as well as the pituitary, the hypothalamus, and the thalamus all become affected once the virus gets to the brainstem. This just gives you a review at postmortem. We go in to collect the trigeminal ganglia, as well as we try to capture most of the cranial ganglia. to see if we can learn how the virus traffics. So, for example, with Beth Griffin's case, she sustained an ocular splash. The trigeminals are tri because there are 3 branches of them. One of those branches subserves the eye and the forward compartment of the eye and the eyelid. And the other 2 serve the mandible and the maxillary regions of the face. And so we frequently begin a postmortem examination looking at the trigeminals to see if they've either subsequently become infected from brainstem virus or if they've, in Beth Griffin's case, if they were the primary site of infection on the way to the brainstem. So what kinds of symptoms do we expect from these centers being involved in the virus infection? And these are the ones we expect and the ones that we see. Nausea, vomiting, and this is the big one where the patient frequently gets shuttled over as potentially having flu, and we're going to treat flu or food poisoning first before we move on to a risk group 4 pathogen. Nystagmus, eye movement, loss of balance and altered gait, and then diabetes insipidus, as I mentioned, and that would be as the pituitary becomes infected by the traverse of the virus, the sinusitis, and of course an ascending paralysis. The lower limbs become involved, and sometimes there's a bilateral paralysis, sometimes it's a unilateral paralysis. But generally, it's a bilateral paralysis. The bladder becomes paralyzed, so urine is retained. And then ultimately, the diaphragm becomes paralyzed, and the person can no longer sustain respirations. And in one case, where an individual lived for over a decade after diaphragmatic arrest, we see the swallowing and the eyelids affected. So again, it's an ascending paralysis. In looking at the brainstem of one of the fatal cases, you can see that there's a herniation from the brainstem that compresses the cerebellum, and you can see the swollen stalk of the pituitary gland, and that, of course, becomes associated with diabetes insipidus. So it's very difficult to get an MRI or a CAT scan with a person that is infected with B virus, similarly to what you would expect if someone's infected with Ebola. You don't like to take patients around that have risk group pathogen and risk group 4 pathogen infections. And so we felt very fortunate at the time to be able to acquire these images. Now that we know a little bit more about the disease, it's sometimes we note that physicians are a little less willing at this point to explore new MRIs or CAT scans. And so that's unfortunate because I think we could gain a great deal more information about the disease and about what's happening. And of course, you can see the normal pituitary in this slide, and you can see the thickened pituitary stalk when infection progresses. So nystagmus is one of the effects that people report seeing first at the level of care given it by the emergency department. And that indicates to us that the ocular motor nerves, or cranial ganglia 3, are becoming affected. Now, at this point, if the person was sustained zoonotic B virus from a bite on the hand, that gives us the information that the virus does move up the brainstem and starts to affect cranial ganglia as well. So with the ocular motor ganglia affected, we do see quite a bit of nystagmus, abnormal eye movement, and really a loss of control of eye jerking motion. So this is very distressful for patients. And at this point in time, frequently the patients, I think really fall through the cracks a bit because there are a number of things that can cause these types of symptoms, but at the same time, disease has rapidly advanced, and it's very imperative at this point to get the persons or patients onto ganciclovir as quickly as possible, if there's any hope to prevent mortality. So I'm going to tell you about another little case that we experienced, and that is an individual who survived the initial infection with B virus, was infected, and I showed you previously 10 years of serologic data on that patient, was infected in 1980, but in 2004 was now 41 and came to his ophthalmologist with decreased vision and pain in his left eye for 3 weeks. So this individual bore the pain for 3 weeks. His past medical history was remarkable because he was one of only 23 known survivors at the time from B virus. And he was diagnosed, by the way, late, about 10 years after his infection, his primary infection. So at this point, the ophthalmologist decided to send this individual to a retinologist who was quite sharp and was able to see very quickly that there was some retinal damage. And furthermore, that damage appeared to have occurred over a number of years, but now seemed to be very acute. You can see some scarring on the supraperiphery that presumably is from previous reactivations. And during this period of time, the right eye was normal. I'm not sure which screen you can actually see my cursor on. Can you see the cursor at this point?
Yes, we can.
Okay, great. So you can see the right eye looked fairly normal at this presentation in 2004, but the left eye had degenerated. The previous visual acuity was 20/40. And it had now dropped to 2400. There were sclerotic vessels, and again, lots of evidence of scarring. So the patient was subjected to a vitrectomy, vitreous fluid was collected and sent to us and was analyzed by PCR and culture and was heavily infected with B virus. We were not able to culture the virus, but we were able to show that the levels of viral DNA were exceedingly high in the vitreous humor. So the individual was put onto ganciclovir. From that point, the infection seemed to get a little bit better. The individual was discharged after about a month on Valtrex, but found Valtrex very expensive and ceased to comply with the regimen in about October. So 6 months later, in January, we get another call that the other eye, the right eye, was now infected or problematic, was showing symptoms. So we collected vitreous humor from the right eye. Again, that was even more laden with virus than the left eye. And things quickly deteriorated from this point with dilated vascular trunks, macular hemorrhage, a detached retina, and so the individual was again put on ganciclovir, and then eventually, once the vitreous returned to a negative status, was discharged on oral ganciclovir. And as far as I know, and I do speak with this individual on a fairly regular basis, is very compliant with the oral ganciclovir. So that gives you a little bit of a bird's eye view of the kinds of things that you look for in patients. But I think probably more important than those things are looking for much earlier signs. And the earliest signs that we can look for can occur within 7 to 10 days, and that's the presence of antibody. But we're very interested in exploring why B virus is doing this, because it is a simplex virus. Humans are very closely related to macaques genetically, and this virus is very closely related to herpes simplex. So why is this virus behaving so strangely in a closely related host? If we can begin to understand that, we can perhaps begin to understand something about cross-species infections. When a virus gets into another species that has not had the opportunity to co-evolve with, there seems to be a loss of the ability of the host to respond with many of the protective innate responses that can defend the human. And so we are investigating many of these responses, and much to our surprise, we're finding that many of the innate immune responses that we expect to see, and that certainly we see when a human becomes infected with herpes simplex, are absent when the individual becomes infected with B virus. And even if they've had a previous history with herpes simplex, we still see a loss of these innate responses. So B virus is not curtailed by innate responses as effectively in humans as it is in macaques. With macaques, we do see many of the innate responses intact, and we see that in macaque cells, there is clearly an adaptation to control this virus and to minimize replication. And that's absent in humans. The adaptive responses do seem to be very hardy in the macaque, but they're very weak in the humans. We can detect humoral antibody, and we can detect interferon gamma from CD4 and CD8 T cells. We can also detect specific cytokines and chemokines, but to link those specifically with an infection requires a great deal more research. So we also investigate the inflammatory responses and NF-kappa B responses that pro-inflammatory responses that can minimize infection and cause the cells that are infected to commit suicide and then prevent the further spread of the virus. So we have studied, but at the point where we lose some of these defenses, we are really in a world of hurt. Can you see the presentation at this point? I think we're loading back up on the PowerPoint, but let me, while we're waiting for that, if we lose the ability to control the infection and we have to rely completely on the adaptive immune response, there is a period of about 7 to 10 days when the virus really has the liberty to replicate aggressively in the body. And so we'd very much like to discover ways that we can stimulate the adaptive, not the adaptive, I'm sorry, the innate immune system in individuals that become infected with the virus. And part of that is learning how to stimulate other aspects of the defenses, to stimulate, for example, the pro-inflammatory defenses, such that we can then engage the adaptive system on the correct timetable. What I was going to show you, and I'm not sure we are going to be able to get the PowerPoint back up, but there are very effective antivirals, and those antivirals include Valtrex and acyclovir. And I'm frequently asked that whether or not these drugs are toxic. In fact, we are very lucky because these drugs are virtually non-toxic. It is a pain to have to take them on a daily basis. But we have an armamentarium with ganciclovir if symptoms begin to worsen and if we have had frequent reactivations. But we also have some fluorinated uracil derivatives that one of our investigators here in the Atlanta area was able to provide for us so that we would test that both in cells as well as in animal models. And those drugs are actually extremely potent at very, very low levels. And because they're extremely potent at very, very low levels, the toxicities that ordinarily are associated with these drugs are thought not to be a problem if we ultimately have to depend on either of these compounds. The other thing that we've recently discovered is that there are drugs that we can use, such as flavonoids, that actually enable us to reduce the amount of Valtrex or acyclovir, and perhaps even ganciclovir that patients have to take. These flavonoids are basically phosphorylase inhibitors and can do a great deal in terms of stimulating specific innate responses that can effectively be used to modulate the amount of virus that is replicating. So with that, I was going to show you all the people that have contributed to this work, many investigators over the years at multiple institutions, Georgia Tech, Emory, certainly at Georgia State University, Texas Biomedical, Oklahoma State University, as well as the military who helped us convert a lot of the data to algorithms that we can use for effective diagnosis and early identification of disease. So with that, since we don't seem to be able to get the PowerPoint back up and running, I'd like to ask if there are any questions.
Any questions? For those who actually dialed in, you can use the chat bubble to type in your questions if you do have some.
I have a question.
Okay.
So, you know, after there's an exposure and blood is collected from the animal, how accurate are those tests? Is it, you know, and can you tell that the animal wasn't shedding virus pretty accurately, or no?
Yes, we can tell if the animal is shedding virus. There's a couple of issues that I should mention. Can you see the slides again?
Yes, you can.
Okay, great. The issues are that under the best of circumstances, when virology specimens are shipped, there is only a 60% chance that they will reach the diagnostic lab in a viable state. And so we rely on PCR for those samples. We don't normally perform PCR because PCR is not going to tell you if there's infectious virus present. But if we do have problems or we suspect that the integrity of the sample has been compromised, we will run PCR just to give you a sense of whether there's DNA present in the sample. The tests are very sensitive, very specific, but serologically and virologic. So diagnostic samples are extremely helpful and important. And in the more recent cases, 2008, we've been able to see antibodies in new patients as quickly as 10 days post-exposure. Please feel free to email us at any point in time or call with any questions that may come to you later. Thank you.
All right. Great. Thank you.